Antibiotics. Our body is composed of cells, and this human cell number is counted as 10 trillion cells in adult body. And conventional wisdom said for a long time, we have 10 times more bacteria cells than our own cells. But recently, the scientists recounted carefully, and we found more cells in our bloodstream. So a lot more red blood cells were found than we expected. So about 70% of our cells are red blood cells. Now we count human cell number as about 30 trillion cells. Bacterial number was a little reduced in recalculation, so that's also about 30 trillion cells. So now we believe the human cell versus bacterial cell ratio is about 1 to 1. In terms of the genomic diversity, our cell has only our own genetic information, but there are a lot of bacteria as a mixture. So altogether we call them as microbiome. And the gene diversity in bacteria altogether, that's more than 100 times richer source than our own genomic information. Now, when the bacteria balance is preferred to probiotic, then our body is in a balanced state and we keep the harmony together with this microbiome. But in bad cases, the bad bacteria or pathogens dominate, then we can get disease. Then what can we do? Well, to remove those bad bacteria, maybe you can take antibiotics, but antibiotic can kill probiotic too. After that, maybe you can feed prebiotics, which is a nutrient for the probiotics. Then probiotic prevalent, the new balance can be recovered. Bacteria is a single cellular organism. They don't have a nucleus, but they have genomic information in DNA. And also they have a plasma membrane inside and cell wall outside. Sometimes they have a flagellum, and using this flagellum, they can swim and move around. Many bacteria are not harmful to human beings, but some bacteria, they can cause disease. Then we should fight against this infected bad bacteria. When we have bacterial infection, our drug choice is antibiotic. Here the bio means this bio is the bacteria. So antibiotic is the drug against bacteria, is antibiotic. How do we measure? First of all, we grow bacteria cell and then drop those drugs. Here it is antibiotic. And then if the antibiotic kills those bacteria, then they can make this empty space. If the antibiotic effect is stronger, then we can see more empty space like this. So in this picture, by measuring those empty space, we can give the rank. This is number 1, and these are number 2, number 3, kind of activity measurement. What is the first ever antibiotic? Many people believe that that's a penicillin, discovered by Alexander Fleming. Well, historically speaking, this is not exactly the true because there were another drugs like sulfa drugs has been used. But definitely penicillin's impact was huge. That's why many people has the impression that the first most effective antibiotic is a penicillin. Now here is the story of a penicillin discovery. Fleming was culturing Staphylococcus, so toxic bacteria, and one day he found the culture was contaminated by the fungus, Penicillium colony, and he observed that around this fungus there are clean area. That implies that this fungus secreted some magic molecule which kills the bacteria. So this is the beginning of penicillin discovery. Sometime later, the magic material was elucidated as penicillin. There are many kinds of penicillin followed. That's why this penicillin is called as a penicillin G. And it was found that penicillin is effective at killing gram-positive bacteria. but the effect was much lower against the gram-negative bacteria. Gram staining method was developed by Dr. Graham. That's why we use the gram staining's G with a capital G because that's a human's name. And he tried many different kind of the dyes to stain bacteria. And one day he found that crystal violet stains only some type of bacteria, in this case, the Staphylococcus. And some bacteria were not stained by this. So he gave the classification of bacteria gram-positive and gram-negative. And penicillin showed activity much better in gram-positive bacteria. Long time later, it was found that the difference between gram-positive and gram-negative in their cell wall structure. Both of those bacteria, they have a cell membrane inside, but gram-positive bacteria has a very thick peptidoglycan layer, and gram-negative bacteria, they have very thin peptidoglycan, and outside of the peptidoglycan layer, there are another membrane, so double membrane structure they have. Now it was discovered that the penicillin's target is peptidoglycan area. That's why penicillin does not have much effect on gram-negative bacteria, but much stronger effect against gram-positive bacteria. Cell wall is composed of peptidoglycan structure. So these are the peptide, and then these are glycans, carbohydrates. So they should be connected in the middle to make the strong structure of cell wall. And there is an enzyme named as a transpeptidase should connect this peptide. Then it can make connected network of a peptidoglycan structure. Now we can see the mechanism of penicillin. Penicillin, all the penicillins has this four-membered ring of cyclic amide structure. Cyclic amide we call lactam. And from here you can count alpha and beta. Four-membered ring of a lactam is a beta lactam. If it is a five-membered ring, it can be gamma lactam. So four-membered ring is relatively unstable chemically. So when it binds to the transpeptidase enzyme, then this four-membered ring can be broken and open, and then that can be connected to transpeptidase. This reaction is specific and also it is a covalent binding, so that kills the activity of this enzyme. So this enzyme cannot do their function to make the connection of peptides. So this is the mechanism of penicillin. After discovery of the structure of penicillin, so it was found that this beta-lactam structure is a key part, which is connected to five-membered ring here. And then it was also found that this side chain can be changed with the different structures. By changing this motif here, and then improved activity was developed. So through structure-activity relationship, the new derivatives of penicillin was synthesized, and they have a better effect and also active against resistant bacteria kind of things. So penicillin V or amoxicillin is kind of improved penicillin structures. Further derivatization made new class of compounds. With a six-membered ring, it is called as a cephalosporin, and sulfur was changed with the carbon is a carbapenem. This is a new class of second, third generation of beta-lactam, the antibiotics. They have a strong effect, also broader spectrum, even can attack gram-negative bacteria. Another kind of transpeptidase inhibitor, vancomycin, looks like a huge molecule. Target is also the cell wall synthesis, but it does not bind to enzyme itself. It binds to cell wall structure before they are connected together. That's why when vancomycin binds to the cell wall precursor, now the transpeptidase cannot bind. So this is kind of a competitive inhibitor against the transpeptidase. Penicillin and vancomycin targets the cell wall synthesis. But later stage, it was found new class of antibiotics. Some of them attack DNA synthesis. The enzymes involved here, DNA polymerase or RNA polymerases, they are different from human enzymes, so there could be some selectivity achieved. Also, the protein synthesis, the machinery, ribosome is also different between human and bacteria, so we could get some selective ribosome inhibitor for antibiotic. Some metabolic pathways, human being, we don't use it because we take those the folate by food uptake, but bacteria need to synthesize biochemically. So if you can block those metabolic pathways, we can selectively kill antibiotics. So the key point for generating efficient antibiotic with a low side effect against the human being is the selectivity. The drugs, the antibiotics should attack only bacterial machinery, not human machineries.